I'll let you in on a little secret. This says E. coli, but it isn't because I couldn't be trusted. In today's video, we're going to try and answer a very simple sounding question, which turns out to be a bit of a stinger. How many bacteria are in this bottle of E. coli? Now to do that, we're going to need a whole host of lab techniques, so we're going to take you through them one by one to try to get to an answer at the end. Let's do it. Now, if a liquid sample has just one bacterium in it, and we put that sample onto an agar plate, and the bacterium is allowed to grow and reproduce, it would develop into a single visible bacterial colony. This gives us a method for working out how many bacteria are in a sample. If there were only a few bacteria in there, then a few bacterial colonies would develop and could be counted, showing how many bacteria we had in our original. But if the sample has too many bacteria, then the colonies all join together and you can't count them. So to get round this, we can dilute this sample again and again, and a sample of each dilution grown on agar plates. That's what we call a serial dilution, and eventually we'll have diluted the original sample enough that one of them should have a number of separate colonies that are countable. Then we can do some maths, scale this up to work out how many we had in our original sample. Still with me? First things first though, we're going to need to be working on what we call aseptic technique. Essentially, everything has to be sterile to protect our work from contamination and protect us and everyone else in the lab from infection. So that's job one. Okay, so we've given our colonies a few days to grow, and now we can choose a suitable plate to run our counting. Ideally, we want somewhere between 30 and 300 colonies on the plate. So this one, 10 to the minus 5, has too many to count. This one, 10 to the minus 6, looks good. And I've already counted exactly 140 colonies. That was in our 0.1 milliliter sample, which is equivalent to 1,400 bacteria in 1 centimeter cubed. 6 1 in 10 dilutions were made from the original sample, which means we need to multiply our 1,400 by 10 to the power 6. So that's 1.4 billion bacteria per centimeter cubed in here. So that's how we answer our question. 1.4 billion bacteria in every milliliter in this bottle. Thank you for sticking with us. Do use the protocol in the lab when you need to. There's a link to a written version in the description below, and we'll see you next time.